For this experiment, all you need is this: drinking water, a little container to collect your sample, a measuring spoon that measures half a teaspoon, diluted dish soap. We dilute 1 tablespoon of dish soap in half a cup of water. We use meat tenderizer as a source of proteases to digest the proteins. And you will also need isopropanol it's isopropyl alcohol, 70% to 95% that you can find in the store. The importance here is that it has to be ice cold. For the artistry part of this experiment, instead of using a tube to perform the DNA extraction, we will use this glass container. We need a container that is clear so we will be able to see the DNA precipitating, clumping. So let's start having fun. So, first, half a teaspoon of water that we put in this small container. First, we suggest that you gently chew the inside of your cheeks, very gently, just to collect a few more cells from your mouth for about 10-20 seconds. Then we use this half teaspoon of water as a mouthwash. We don't drink it, we just use it as a mouthwash. We will then spit it out in the same container, and this will be our sample. And we spit it back out. This is now our DNA sample. So now in this container, I have a water solution with cells from my mouth. And this is our sample. We will extract DNA from this sample. So the first step is breaking open the cells because the DNA is in your cells. So to do that, we use detergent. We put our sample into our tube. And remember, we have cells in this water solution. We will use about half a teaspoon of detergent. The skin of your cells is mostly made of fat. The detergent will help break open the cells. So now we mix our cell solution with the detergent by slowly inverting 5 times. Now we have our cell solution mixed with the detergent. What we want to do is also add proteases. So we want to destroy the proteins. We use meat tenderizer because it has salt that will allow the debris, the cell debris, to clump. And it mostly also has bromelain, which is a protease. And a protease is an enzyme that will digest proteins. So I will just add a pinch of meat tenderizer that I will mix with my detergent and cell. And same thing, slowly I will mix by inverting slowly 5 times. So now we could let it sit for about 10 minutes, either at room temperature or at a slightly higher temperature, while we're opening up the cells and destroying the proteins to kind of free the DNA. What I like to do at this stage is to actually put this mix of cells, detergent, and proteases in a warm water. So what I've done is I've taken just a cup filled with water that I've microwaved for about 30 seconds, and I place my tube in there and I let it incubate for about 10 minutes. At this stage we have our mix of cells that have been broken open with the detergent. Proteins have been digested, so we have our DNA in solution. What we want to do now is separate the DNA from everything else. So now time to precipitate the DNA. Right now you can't really see it, but we have a collection of cell debris, some white clumps. So we have our DNA in solution, so now we want to precipitate it. We want to separate everything else from the DNA. So because DNA is not very soluble in alcohol, we use the ice cold isopropyl alcohol. So what we're going to do, we want to make sure we tilt a little bit the tube and slowly add so we can actually start seeing at the surface DNA precipitating. So now we should be able to see the DNA. In this black background, we can see that the DNA is starting to precipitate. So we will wait about 5 more minutes. I will then start mixing slowly the isopropanol with the DNA. There it is. What a beauty. You can better see it in this black background. The stringy this is the DNA. So we have just extracted DNA. This is my DNA. How cool is that? This is DNA. This is my DNA. And you can do the same at home. How cool is that?